Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
Welcome to one of the Q4.HSC exam question videos. In this video we're going to cover this exam question, which comes from the DNA to protein chapter. What I'll do in a second, I'll read the actual question. Once I have the question, you get about 5 seconds to pause the video. Once you've paused the video, attempt the question and press play when you're ready. The question itself, 2 parts to this question. A, how could a mutation in DNA affect polypeptide production? That's worth 3 marks. And B, how could a change in the polypeptide affect the cell activity? And that's worth 2 marks. So pause the video and when you're ready, press play. Welcome back. Right, for the first one, it's worth 3 marks. It's actually worth quite a decent chunk for that kind of question. So how can a mutation in DNA affect polypeptide production? So the first thing you should do is you should obviously link DNA to protein production. Quickly describe how DNA leads to protein production. You don't have to go into much detail because that's not what the question is asking. Don't talk about translation, transcription in detail. Just say DNA can be made into amino acids, codes for amino acids, which then make your polypeptide chain. So how DNA leads to polypeptide, not protein. And then you would say what happens when there's a change in the DNA sequence. So a change in the DNA sequence, what that does. And obviously link that change in DNA sequence to change in the polypeptide. So how, if we change the DNA through mutation, how a new polypeptide gets produced. What you can also do, this would be quite good, is talk about what the different types of mutations. You know, we said insertion, deletion, everything else. That would be a good point to mention those as well. Just so you can show the marker that you know what you're talking about. Right, so what I wrote first is a polypeptide is a chain of amino acids that was coded for by a certain base sequence in DNA. Right, that was the first part. DNA leads to polypeptides. Here, that's what I did here. And that'll get you marked because you need to talk about that. Then the second part is if the base sequence is changed through either insertion, deletion, or point substitution mutations. So here these different types of mutations can change the base sequences. Then the base sequence will code for different amino acids. So now we've said then, okay, well, amino acids, DNA codes for amino acids. And if we change the actual base sequence, that means we're going to get different amino acids. And after that, I'm going to wrap it all up. So the code this will code for a polypeptide chain that is different from the original polypeptide chain. And this change is due to the mutations. Right? So the different amino acids are coded for by the new DNA. And obviously, all those amino acids will link together to form a polypeptide chain. And the ultimate polypeptide chain that will be produced will be different from the original one. And the reason why is because of the mutations. Right? So here we've gotten all of 3 marks. We've said what DNA does, it codes for polypeptides. What happens if we have a mutation? That means the amino acids that are the building block of polypeptides get changed. And if the amino acids get changed, that means the polypeptide will also be different. And that's your 3 marks here. Now the second part, how could a change in the polypeptide affect the cell activity? So what we should have written here is you should have talked about that polypeptides are linked to proteins, so polypeptides make proteins, and that they determine the structure or the function of the proteins. So how they curl up determines the structure and the function of the proteins. If you have a very different polypeptide than the original one, the intended one, that means the protein itself might not work anymore because the structure and function have been changed dramatically, and that would obviously affect the cell. And you can give examples. Enzymes wouldn't work anymore, or you might have the cell membrane proteins don't get produced as they should. If you want to give an example, you could give an example too. So what I wrote is a change in the polypeptide sequence will result in different proteins. So these different proteins have different functions and or different structures being produced. So because of the new polypeptides, we have new proteins and they have different functions or structures. It could result in the production of enzymes. This is the example I gave. which are proteins that have lost their function. In many cases they have lost their function, sometimes they have a new function, but in many cases they have lost their function. So if we have enzymes that have lost their function, what could happen is the next sentence: this could severely alter the normal biochemical pathways. If that word biochemical pathways sounds unfamiliar, that's just the idea of all chemical reactions inside the cell. So a cell needs to do chemical reactions to stay alive as well. They need to stay alive, and that's their biochemical pathways. Obviously, if we lose our enzymes in our cells, that means these chemical reactions won't go ahead as usual, and that could be bad for the cell. So this could severely alter normal biochemical pathways that are needed to maintain cell health and could lead to the death of the cell. Because if the enzymes aren't there anymore, because they might have their function changed or their structure changed. What I mean by enzyme structure changed would be denatured. So they might have been denatured, they can't break down substrates anymore. But in any case, that would be bad for the cell. It will lead to the death of the cell. Now, this was one example. Obviously, the same thing happened when it comes to a mutation led to blue eye color. And the blue eye color obviously isn't something that kills us. But if, for example, we have a mutation with enzymes, often that's bad for us. Mutations, some other things might not be that bad for us in general. But sometimes, even with enzymes, when they get a new function, we talked about nylon bacteria in video 12. They had an enzyme which had a new function which allowed them to break down nylon. So it doesn't always have to be bad. Mutations aren't always bad, but often they are bad for the actual organism with the cell. Right, so here we get 2 marks, just for quickly mentioning that, if you have a different polypeptide being produced, that means we're going to have different proteins that have new functions and structures being produced. That gets you a mark. And then linking that to how the selectivity could be changed. So for example, we said, okay, well, enzymes are proteins. These enzymes might have lost their function because of their new structure. And that means that the cell's chemical reactions will stop and the cell will die. That gets you a mark as well. That's worth 2 marks out of 2. And where does the question come from? It comes from this here. Students will process information to construct a flowchart that shows the changes in DNA sequence can result in changes to selectivity. So from DNA to change in selectivity. Here they broke that into 2 parts. First they said, how could change in DNA lead to a change in polypeptide production? And then from polypeptide production to a change in selectivity. So it's basically this dot point, just broken down into 2 bits. But I hope that was useful.
Thank you for watching.